Okay, in the previous lecture, I mentioned about simple cuboidal epithelial tissue. In this lecture, we are going to talk about simple columnar epithelial tissue, its various modifications, where those modifications are found, and what's their role in those places where they are found. So firstly, simple columnar epithelial tissue. What is that? That's an epithelial tissue. Since it's simple, the cells are arranged in single layer, and since it's columnar epithelial tissue, the cells that are arranged in single layer are columnar or long. Okay, so this tissue has three major modifications. Out of those, the first modification is ciliated. So you have ciliated simple columnar epithelial tissue. What is that? That is your columnar tissue. That is present on the basement membrane. Then there's your round nucleus present at the basal region. I mean, that's your normal simple columnar epithelial tissue. The modification is that now these cells contain cilia. Oh, no. So, on the free surface of the simple columnar epithelial tissue, if cilia is present, then that's your ciliated simple columnar epithelial tissue. Now, what's the significance of the cilia? The cilia that is present on the free surface are none other than your kinocilia. This part, kino, comes from the word kinetics, which deals with movement. That means the cilia that are present in ciliated tissue are motile, and the type of movement they show is a rowing movement. What is rowing movement? Basically, it means to say that the movement that takes place is unidirectional, or it takes place in one particular direction. How? Okay, imagine you are on a boat and then you are on a lake. You have two rows to row the boat. You start rowing the boat. How do you do that? You take the two paddles and push it backward, which pushes the water backward as well, and the boat moves forward. Then you lift those paddles up and then push it back again, and the same process takes place again and again and again, and the boat moves forward. Similarly, in kinocilia as well, the cilia beats in one particular direction. These are your cilia present on the columnar cells, and they beat in one particular direction. Now you may be thinking, but the cilia are moving backward as well. Yep, associate or just relate the backward movement of the cilia to the lifting of the paddles. So it does go backward, but it doesn't have significance or much force being exerted in the backward direction. The force is being exerted when it beats in forward direction. That's it. Where is it found? It's found in the fallopian tube. Fallopian tube. Here's your uterus. And then here's your fallopian tube. Okay. The fertilization of the sperm with the egg from the ovary takes place in the fallopian tube, and the formation of zygote takes place. This zygote right here needs to be transported from fallopian tube to the uterus. And that job is done by none other than your ciliated simple columnar epithelium. How? The cilia or the kinocilia present on the free surface of this epithelium starts to beat, and this beating motion transports the zygote present right here into the uterus. That was your first modification. The second modification is Bros border. So Bros border epithelium. In Bros border epithelium, again, you have your simple columnar epithelium, right, which is present on the basement membrane, which has nucleus on the basal region. In case of ciliated epithelium, on the free space, cilia was present. Similarly, in case of Bros border epithelium, the free surface, not the space, sorry, surface contains Bros border. Bam, 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 wiggle, 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 wiggle. Now, to make it clear why it is known as Bros border, I will extend this diagram furthermore. And from that, you'll clearly understand why it is called so. So this is your epithelium, right? I'm not gonna do anything. Just look here. And then what does it look like? It looks like a toothbrush. I put paste on it as well. I mean, you can't brush with it, but at least you will know it about it for the exam. Anyway, so the border looks like the border of the bristles of a brush. That's why it's known as Bros border epithelium. What's the significance? I've already mentioned in the previous lecture. Having Bros border increases area. Increase in area increases absorption. So where is it found? It's found in those places where absorption is necessary, like your intestine. How is it present? Let me show you. So you know this much, that your intestine is lined by something known as villi. What are villi? Villi are finger-like projections that are present in your intestine, which increases the surface area, which in turn increases the absorption capacity. What is this yellow thing that I'm drawing inside of the finger-like villi? That's your lacteal. That's where the monomers of fat goes during absorption. Villi is also supplied with capillary. So this is the arterial part of the capillary. Bam, 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 bam. And then here's your venual part of the capillary. Boom, boom, boom. This is where the absorption of monomers of fats and carbohydrates take place. Now, this villi is lined by epithelium. Tuck, 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 tuck. Again, I have mentioned it before as well. If you find the pace to be slow, please watch it at a higher pace. I ain't gonna stop you. I ain't gonna come to your home and start slapping the shit out of you. No, no, no. Just watch it at a higher pace. And this epithelium that surrounds your villi is none other than Bros border simple columnar epithelium. And the Bros border of that particular epithelium in this particular case is known as microvilli, because it's on villi and they are finger-like projection as well. So these are small finger-like projection on large finger-like projection. So these are microvilli, smaller villi on villi. What's the benefit? Increases surface area for absorption. That's the benefit. Okay, next is, and the final modification is your goblet. Oh, let me use the right color. Is your goblet cell. Goblet cells. So, goblet cells are called goblet cells because they look like, drumroll, goblet cells. I'm so sorry. They look like goblet. And what's goblet? Goblet is a type of glass which looks something like this. It has a wide apex and a narrow base. And the goblet cells looks exactly like this. You can fill this with water or maybe wine or beer. I don't drink any of that. I hope you don't as well. This is what goblet cells look like. It has a wide apex and a narrow base as well. Boom. Since it's a columnar cell, the nucleus is present at the base, and the apical region contains lots and lots and lots and lots of vesicles. And you know, if these are vesicles, vesicles have to be filled with something, and the thing that fills vesicle, that is filled in these vesicles, are mucus, which gets released outside. So where could these goblet cells be found? Everywhere where there's mucus, these goblet cells are found. Throughout your gastrointestinal tract, you have mucus lining in the inner wall. So throughout those lining, you have goblet cells. And boom, that's your simple columnar epithelial tissue. Let me revise it for you. There are 3 modifications. First is ciliated modification, where the free surface of the tissue contains cilia. The cilia is motile, so also known as kinocilia, and it shows a rowing movement, that is, it beats in one particular direction. What is its significance? This epithelium is present in the fallopian tube. After the fertilization, zygote formation takes place, and this zygote formation takes place in the fallopian tube. This zygote needs to be transported to the uterus. So the function played by the epithelium is transportation of this zygote to uterus. Then second is bross border epithelium, where the free surface looks like the end of or the border of the bristles of a bross. So it's known as bross border. It increases surface area for absorption, and it's found surrounding the villi throughout your intestine. What's the significance? Helps in absorption. And finally, you have goblet cells, which are secretory cells or glandular cells or gland cells. They are found throughout your gastrointestinal tract, and they produce and release mucus, forming the mucus lining of your digestive system. So the significance is secretion. Whew, we are done. Ta-ta!